Let us now see what all different kind of bonds are found in a DNA molecule. So as we have seen earlier, I told you that bases are attached to each other by phosphodiester bond in a polynucleotide chain. Then I also told you that within a double helix, in helix, two bases, for example, when A pairs with T or G pairs with C, are held together by hydrogen bonds. These are the bonds that are required for maintenance of polynucleotide chain and holding up two polynucleotide chain to form a helix. But besides this, there are some other bonds also which are present in a nucleotide, and they serve as high energy bonds. And these bonds which also serve as high energy bonds are phosphoester bonds and phosphoanhydride bonds. And as I told you previously, there is another phosphodiester bond, and of course hydrogen bonds. So hydrogen bonds we have already talked about in the other lecture. We will concentrate on these three bonds which are primarily present in DNA. So let us first see what is a phosphodiester bond. Phosphodiester bond is formed between 3' sugar and a phosphate group of a nucleotide. How is it formed? Suppose this is a base. I am, for example, making a pyrimidine base which has N here, and then it binds to a sugar. Now this is 1' carbon, this is 2' carbon, 3', 4', and CH2OH, this is 5' carbon. Suppose this is the first carbon, and now we have a OH group here, and if we have another base, say for example it is a purine base, and now we have let us say we write it as minus. So what will happen? This water molecule will move out and a bond will be formed which will be now ester, as we know, Ester is, in chemistry you would have studied, is represented as R oxygen R dash. These R are two different functional groups. So one functional group is this. Suppose this is R, and the other functional group is this. This is R dash. So R dash and R are connected by this bond, and see again there is another bond here. It can also be an R and R dash. So because there are two esters, this bond is known as phosphodiester bond. Fine. So this is a phosphodiester bond which is responsible for continuance of a nucleotide chain within a DNA molecule. So after this, another important bond that we find in a polynucleotide chain is not exactly polynucleotide chain but in nucleotides, and that is known as phosphoester bond. Now the phosphoester bond is, suppose this is a base and this base is attached to a sugar. Okay. Now we have O, and then first phosphate molecule is attached here. Now this phosphate and this whole thing acts as an ester. So this bond is called as phosphoester bond. Now you see that the first bond of phosphate that is joined to the carbon molecule of a sugar is a phosphoester bond. This is a high energy bond. When it is broken down, it produces a lot of energy. But beyond this, if you add another phosphate molecule, so now it becomes nucleotide diphosphate or nucleotide triphosphate. For example, if it is ATP or CTP, then these two bonds that are formed are called as phosphoanhydride bonds, and these three bonds are very high energy bonds, and that is why when ATP is used as an energy point, these bonds are broken and the amount of energy that is liberated is used for carrying out a reaction. So primarily there are four types of bonds that are found in a nucleotide, and they are phosphoanhydride bond, phosphoester bond, phosphodiester bond, and hydrogen bond that hold together two polynucleotide chain in a helix. Thank you.